welcome to our channel Medalixer. This is the next video on physiology. So in this we are going to discuss about electrophysiology. The objectives of this session include Gibbs Donnan effect, resting membrane potential, tonic potential, action potential and conduction of impulse across a nerve. First we are going to see about what is Gibbs Donnan effect. So what is Gibbs Donnan effect? So Gibbs Donnan effect states that presence of a non-diffusible ion on one side of the membrane separating two solution will affect the distribution of other ions to which the membrane is permeable. So let I say with an example. So let this be a semi-permeable membrane separating two solutions A and B. So the presence of non-diffusible ion on one side of the membrane, on either one side of the membrane. So let this be a non-diffusible ion. So this presence of non-diffusible ion will affect the distribution of other diffusible ions. It will affect the distribution of diffusible ions across the membrane. Okay, to explain Gibbs Donnan effect, we shall take an example. So the example is our extracellular fluid and intracellular fluid which is separated by the cell membrane. As we all know, our cell membrane is semi-permeable which is made up of lipid bilayer. So what are the ions present in extracellular fluid and what are the ions present in intracellular fluid? So we shall take an example that in extracellular fluid we'll take only potassium and chloride. Okay, so in extracellular fluid there presents potassium ions and chloride ions. And what are the intracellular ions present in intracellular fluid? The ions present in intracellular fluid include potassium ions, chloride ions, and also there is then negatively charged proteins. So as we all know, intracellular fluid is rich in negatively charged proteins and other organic negatively charged compounds like sulfates and phosphate compounds. We'll take an example of proteins. Okay, so proteins are negatively charged, and as our membrane is semi-permeable, the proteins are not permeable to our cell membrane. So in this our example, the protein is the non-diffusible ion. Okay, so protein is a non-diffusible ion. Okay, so the same example here too. So this is an extracellular fluid and intracellular fluid which is separated by our cell membrane. Our intracellular fluid consists of potassium, chloride and proteins, which is a non-diffusible negatively charged ion, and extracellular fluid contain potassium and chloride. So normally to maintain electrical neutrality, the concentration of cations and anions on both sides of the membrane should be equal, right? So normally what it should be? So normally the cations of ECF should be equal to the cations of ICF. So the concentration of cations of ECF and ICF should be equal. And also same, anion concentration of ECF should equals the anion concentration of ICF. This is what normally it should be to maintain electrical neutrality. But so applying this, what is here? So cations of ECF. What are the cations? So here the cations of ECF. What are the cations of ECF? It is only potassium, right? So from our example, we have potassium. So potassium concentration in ECF should equal to the potassium concentration in ICF, as this is the cation of ICF. And also by the anion concentration, so the chloride concentration of ECF should equals to the chloride concentration of ICF. And what are the negatively charged ions present in ICF? It is chloride and protein. So it should be equal to the chloride concentration of ICF should equals to the chloride concentration of ICF plus the protein concentration of ICF. You understood, right? So as protein is also negatively charged, so the chloride concentration ECF should equals chloride concentration of ICF and protein concentration of ICF. So from this equation, we can understood that chloride concentration of ECF is actually greater than chloride concentration of ICF. Right. So as the chloride concentration of ECF equals both the concentration of chloride and proteins in the ICF, so normally the chloride concentration of ECF should be greater than chloride concentration of ICF. So if chloride concentration of ECF is greater than chloride concentration of ICF, there occurs a concentration gradient. There occurs a concentration gradient for chloride from where to where? So from ECF to ICF. So there occurs a concentration gradient of chloride from ECF to ICF. This causes a movement of chloride from extracellular fluid to intracellular fluid. As there is a concentration gradient, this causes movement of chloride from extracellular fluid to intracellular fluid. And as chloride is negatively charged, as if one of the negative charges move from ECF to ICF, it should be neutral, it should be nullified, or it should be neutralized, so that one potassium also starts moving from ECF to ICF. Because of chloride gradient, as chloride moves from ECF to ICF, and to maintain electrical neutrality, potassium also moves from ECF to ICF. Understood, right? So potassium moves from ECF to ICF. So first, earlier we saw that potassium concentration of ECF is equal to the potassium concentration of ICF. But now as potassium move from ECF to ICF, what becomes now? So now potassium concentration of ICF will become greater than the potassium concentration of ECF. Potassium concentration of ICF will be greater than potassium concentration of ECF. So normally we'll say that potassium is an example of, potassium is an intracellular cation, right? Potassium is an intracellular cation. What does it mean? So it means potassium is excess, or potassium is high in concentration inside the cell. Why? Because this is due to Donnan effect. This is due to Gibbs Donnan effect. So how can we relate this to our example of Gibbs Donnan effect? So due to the presence of protein, which is a non-diffusible ion, the distribution of diffusible ions altered or affected, right? So due to the presence of protein, the concentration gradient occurs, and due to the concentration gradient, the distribution of the permeable ions or the diffusible ions get affected. So this is what Gibbs Donnan effect. So from this example, so we can say that Gibbs Donnan effect, so presence of non-diffusible ions, here the protein, will affect the distribution of diffusible ions, here potassium and chloride. So as we see that due to the presence of protein, chloride and potassium moves inside from ECF to ICF. How long this distribution will take place? So Gibbs and Donnan also said that. The distribution of diffusible ion will take place up to equilibrium. When equilibrium is reached, the distribution will be stopped. Okay, so the distribution of potassium and chloride from ECF to ICF will take place until equilibrium is reached. So how can we calculate equilibrium? So this is by Gibbs Donnan equilibrium equation. So what is it? Gibbs Donnan equilibrium equation. What does this say? Gibbs Donnan equilibrium equation. So it states that due to the presence of non-diffusible ion, the diffusible ions will distribute themselves. So that at equilibrium, the concentration of ions on both sides will be equal. How the concentration? The product of concentration of ions on both sides will be equal, which means at equilibrium, so if we take this example, so at equilibrium, the product of concentration of ions on both sides must be equal, which means the product of concentration means the concentration of potassium in ICF into concentration of chloride in ECF should equals to the concentration of potassium in ICF into concentration of chloride in ICF. So this is what Gibbs Donnan equilibrium equation. So due to the presence of non-diffusible ion, the diffusible ions will distribute themselves until equilibrium is reached. So how can we calculate equilibrium? It is by Gibbs Donnan equilibrium equation. What does it state? So the diffusible ion will distribute themselves until the product of concentration of ions on both sides are equal. So can we rearrange this? So how can we rearrange? So we can rearrange this formula and we can say that potassium concentration of ECF by potassium concentration of ICF is equal to chloride concentration of ICF by chloride concentration of ECF. So this is what Gibbs Donnan equilibrium equation. Understood, right? This is Gibbs Donnan equilibrium equation. Okay. Then moving on to the effect of Gibbs Donnan effect, there are three main effects of Gibbs Donnan effect. So what is the first effect? As we already said, due to the presence of protein, there occurs chloride concentration of ECF greater than chloride concentration of ICF. So what happens? Chloride starts moving from ECF to ICF, right? So chloride starts moving from ECF to ICF. As chloride is negatively charged, we said that potassium also moves from ECF to ICF. And not only potassium, as extracellular fluid also is provided with sodium, sodium can also move from ECF to ICF. So from in a cell, as sodium and chloride moves in, both are osmotically active. So as both are osmotically active, they drag in water along with itself, along with it. Right, so sodium and chloride also drags in water along with it. So what does happen? So as long as sodium moves inside and as long as chloride moves, water also moves in. So as water accumulate in the cell, at last it will cause lysis or rupture of cell, which is called as osmotic lysis. Osmotic lysis. So one of the important effect of Gibbs Donnan effect is that it causes osmotic lysis. How? You understood, right? So as movement of chloride cause movement of sodium, which are osmotically active, so they drags in water and cause osmotic lysis. But osmotic lysis shouldn't happen normally, right? So to prevent osmotic lysis, we have sodium potassium ATPase. So as we already done in our first video of transport mechanism, what is sodium potassium ATPase? It is the example of primary active transport. It is an example of primary active transport. And what does it do? It pumps out 3 sodium. It pumps out 3 sodium and takes in 2 potassium. And takes in 2 potassium, right? So as sodium is again pumped out to the extracellular fluid, this effect of osmotic lysis is prevented, right? So what is the main effect of Gibbs Donnan effect? So Gibbs Donnan effect states that our cells have a tendency to accumulate water and can cause osmotic lysis. So but this is prevented naturally by our sodium potassium ATPase. This is the first and the foremost effect of Gibbs Donnan effect. And what is the second effect? So the second effect of Gibbs Donnan effect is that, so as we already said, this is a semi-permeable membrane. This is extracellular fluid, intracellular fluid. As extracellular fluid has high concentration of chloride, it starts moving inside, and also potassium will also move inside, right? Okay. So as initially the concentration of potassium in ECF and ICF are equal, but due to Gibbs Donnan effect, the concentration of potassium in ECF is greater than the concentration of potassium in ICF. So due to Gibbs Donnan effect, there occurs an asymmetrical distribution of ions across the membrane. So there is no symmetry, there is no equal concentration. So potassium in ECF is greater than potassium. So this asymmetrical distribution causes a potential difference to be existing across the membrane. So due to the asymmetric distribution of ions, there exists a potential difference across the membrane. So this potential difference which exists after equilibrium is reached, as the movement will continue until equilibrium is reached, right? So as we already said, it will reach up to Gibbs Donnan equilibrium state. After reaching the equilibrium state, even though there is asymmetrical distribution of ions, so there exists an potential difference across the membrane, which is called as equilibrium potential. What is it called? It is called as equilibrium potential. Please don't confuse with resting membrane potential. It is different. Equilibrium potential. So what is equilibrium potential? So the potential existing across the membrane after equilibrium is reached due to Gibbs Donnan effect is called equilibrium potential. This equilibrium potential is specific for specific ions. It is different for potassium, this is different for chloride, and it will be specific for specific ions, and it is calculated by an equation called Nernst equation. So equilibrium potential is calculated by Nernst equation. So Nernst equation is given by equilibrium potential of ion is equal to RT by ZF ln of ionic concentration outside by ionic concentration inside. So Nernst equation is given by equilibrium potential of ion is equal to RT by ZF ln of ionic concentration outside by ionic concentration inside. So what does equilibrium potential mean? So the equilibrium potential is the potential difference existing across the membrane after equilibrium is reached due to Gibbs Donnan effect. Okay, so this is what Nernst, it is calculated by Nernst equation, and this is Nernst equation. And what is the next effect? So the third effect of Gibbs Donnan effect include, so Gibbs Donnan effect is the main effect for distribution of ions, between capillary wall and interstitial fluid. Do you understand? This is capillary. So as this capillary wall, so as capillary contain blood, so it is blood. So as plasma contains more proteins, than interstitial fluid, more ions will distribute across capillary wall due to Gibbs Donnan effect. So Gibbs Donnan effect is the main effect which is responsible for movement of ion between blood and interstitial fluid across capillary wall. Understood, right? So the three effects of Gibbs Donnan effect include: first, it has a tendency to cause osmotic lysis, but it is prevented by sodium potassium ATPase. Second, so it is responsible for equilibrium potential, generation of equilibrium potential. And third, what is it? It is the main effect for movement of ions across capillary wall. Okay, these are the three main effects of Gibbs Donnan effects. Now we'll move on to our next topic of resting membrane potential. So what does resting membrane potential mean? So from the term itself we could understood. So it is a potential difference existing across the membrane when the membrane is at rest. So the resting membrane potential is the potential difference existing across the membrane when the membrane is at rest. So we should be clear that resting membrane potential is not an absolute value. It is just a relative value. So resting membrane potential is the relative value, means it just tells how far the interior is negative when compared to outside. So it is it gives the relative negativity value when compared to interior and exterior. So resting membrane potential is a relative value. Now about the genesis of resting membrane potential. So what is the main cause of resting membrane potential to be generated? So the resting membrane potential is mainly generated by leaky potassium channels. What are leaky potassium channels? So leaky potassium channels are nothing but they are voltage gated potassium channels which are open at rest. So leaky potassium channels are voltage gated potassium channels which are kept open at the rest. So what does happen? As we already said from our Gibbs Donnan effect, potassium concentration of ICF is greater than potassium concentration of ECF. So potassium has a concentration gradient from ICF to ECF. So what happens is that as potassium channels, through leaky potassium channels, potassium from ICF leaks out to ECF. As there is a gradient from ICF to ECF, so what happens is that, so let this be a cell, this is ICF and this is ECF. As potassium leaks inside to outside, as positive charges leak out of the cell, the interior of the cell becomes negative. As positive charges are lost from the cell, the interior of the cell becomes negative. Related to it, the exterior of the cell will be positive. As potassium is a positive ion, it gets out the extracellular membrane. The membrane which is facing towards the exterior will become positive. This is the main reason how the resting membrane potential is generated. So the other causes of resting membrane potential include, genesis of resting membrane potential include the relative impermeability of sodium. So at rest, sodium is relatively impermeable to the membrane. As we all know, sodium concentration is high in ECF when compared to sodium concentration is ICF. So there is a concentration gradient existing for sodium from ECF to ICF. But at rest, as the membrane is relatively impermeable to sodium, it is not completely impermeable, it is relatively impermeable. So as it is relatively impermeable to sodium, the potassium loss cannot be counterbalanced by the sodium intake. So as the membrane is relatively impermeable to sodium at rest, so the potassium loss cannot be counterbalanced by the sodium. So it also helps to generate the negativity inside the cell. This also makes a reason why negativity gets inside the cell. Another cause for genesis of resting membrane potential include, as we already said in Gibbs Donnan effect, we saw that our intracellular fluid is rich in negatively charged proteins. Our intracellular fluid is rich in negatively charged proteins and other organic compounds. As proteins are non-diffusible, they are not permeable through our membrane. So the presence of proteins inside the cell, which are negatively charged, also helps in increasing the negativity inside the cell, so which also helps in the genesis of resting membrane potential. So now as a summary, so resting membrane potential. So genesis of resting membrane potential is done by three things. So first, mainly resting membrane potential is generated by leaky potassium channels. So leaky potassium channels, what does it do? So potassium moves from ICF to ECF, thereby making the interior of the membrane negative. Second, at rest there is relative impermeability for sodium. Relative impermeability of sodium at rest. And the third cause for resting membrane potential, genesis of resting membrane potential, is presence of negatively charged protein. Presence of negatively charged proteins in ICF. Negatively charged proteins in ICF. Now we can understood that resting membrane potential means, due to these reasons, the interior of the cell membrane becomes negative and the exterior becomes positive. So the relative negativity when compared to outer positivity is actually the resting membrane potential. As there are two oppositely charges, there will be a potential difference existing across the membrane, right? So this potential difference existing across the membrane when the membrane is at rest is called resting membrane potential, and it is a relative value, means it is how relatively when compared to exterior, how relatively negative charge it is inside the membrane it is. So that's what resting membrane potential is, relative value. It is not the absolute value. And the important concept here is resting membrane potential. So as we already saw, there is a leaky potassium channel. So potassium move from ICF to ECF. So as potassium move from ICF to ECF, okay, as resting membrane potential is mainly generated through the leaky potassium channels, which cause movement of potassium from ICF to ECF. So if it continues, if the movement of potassium from ICF to ECF continues, one time there will be no concentration of potassium inside, right? So which means the resting membrane potential will be depleted, right? So to avoid that, so the resting membrane potential, to maintain resting membrane potential, what is happening? So resting membrane potential is maintained by our sodium potassium ATPase. How it maintains? As potassium concentration is high in ICF and more low in ECF, which cause movement of potassium from ICF to ECF. What does sodium potassium pump or sodium potassium ATPase do? As we already said, it pumps out 3 sodium and takes in 2 potassium, right? And takes in 2 potassium. So by this, it restores the concentration of potassium inside the cell. So by sodium potassium ATPase, as the concentration of potassium will be restored in intracellular fluid, so resting membrane potential is maintained by sodium potassium ATPase. But the main cause for the genesis of resting membrane potential is that leaky potassium channels. Understood, right? So this is what resting membrane potential. Resting membrane potential of a neuron or if a nerve is minus 70 millivolt, and resting membrane potential of a muscle fiber, a muscle, is minus 90 millivolt. As we'll be mostly discussing these two as an excitable tissue in our physiology, so we are giving the resting membrane potential value of these two. So now the resting membrane potential. So how shall we calculate the resting membrane potential? So the resting membrane potential is calculated by Goldman-Hodgkin-Katz equation. What is this equation? So this is what the equation. V is equal to RT by F ln of permeability of potassium into concentration of potassium outside plus permeability of sodium plus concentration of sodium outside plus permeability of chloride into concentration of chloride inside divided by permeability of potassium into concentration of potassium inside plus permeability of sodium into concentration of sodium inside plus permeability of chloride into concentration of chloride outside. We don't want to memorize this formula, but we should understood that the resting membrane potential depends on what? So the resting membrane potential depends on permeability of ions. So from this formula we can say that, from this equation we can say that resting membrane potential depends on the permeability of ions, right? And also concentration of ions. We only want this, which is resting membrane potential depends on permeability of ions and concentration of ions. And just know how can we calculate the resting membrane potential. So resting membrane potential is calculated by Goldman-Hodgkin-Katz equation. Don't confuse with equilibrium potential. So equilibrium potential, how we'll calculate? So equilibrium potential is calculated by Nernst equation. Equilibrium potential is calculated by Nernst equation. This is Goldman-Hodgkin-Katz equation. Then we will see about some terms. To understand some terms. So what is depolarization? What is hyperpolarization? So we shall understand these two terms first. First, to understand depolarization and hyperpolarization, first we should know what is polarized state. What is polarized state? So polarized state is nothing but the state of cell membrane. So this be cell and this is ICF and this is ECF. The state of cell membrane at resting membrane potential is called polarized state. So the state of cell membrane when the membrane is at resting membrane potential. So the state of cell membrane in resting membrane potential is called polarized state. So at the resting membrane potential, what is the polarity inside and outside? As we already said, it will be negatively charged inside, right? So we already said it is relative value, how relatively negative charge when compared to outside. So inside the membrane will be negative and outside will be positive at the resting membrane potential. So the state of cell membrane at resting membrane potential is called polarized state. So what is the polarity of cell membrane in the polarized state? Inside negative and outside positive. So by this, can we say what is depolarization and what is hyperpolarization? First, what is depolarization? Depolarization, it is quite easy. So we can give from the term, right? So what is depolarization? So reversal of polarity from polarized state is called depolarization. So reversal of polarity from polarized state. So from the polarized state of the membrane, when the polarity get reversed, then we'll be calling a depolarization. So means depolarization can be caused by, how can we cause depolarization? It can be in two ways, right? What are the two ways? Either it is caused due to decrease in positivity outside or decrease in negativity inside, right? So to reverse the polarity, there should be more negative outside and more positive inside, which means there may be decrease in positivity outside or decrease in positivity outside and decrease in negativity inside. So thereby the polarity get reversed, which will attain depolarization, right? So you understood what is depolarization. So what is depolarization? Depolarization is the reversal of polarity from polarized state is called depolarization. So how can we attain depolarization? So depolarization can be attained by either decreasing positive charges outside or decreasing negative charges inside. And now what is hyperpolarization? What is hyperpolarization? So you shall again draw a diagram. This is a cell, which is cell membrane. This is intracellular fluid and this is extracellular fluid. As we already know, so the interior will be negative and the exterior will be positive during resting membrane potential. This polarity of membrane is called polarized membrane. What is hyperpolarization? So the term hyper, what does term hyper means? Hyper means increased, right? So the increased polarity, so the increase in the quality or increase in the polarized state of the membrane. So how can we attain hyperpolarization? It can be either due to increasing negativity inside or increasing positivity outside, right? So increasing negativity inside or increasing positivity outside. Increasing negativity inside or increasing positivity outside. So now understood what is depolarization and what is hyperpolarization, right? So if we understood what is depolarization, hyperpolarization, so the study of action potential will be very easy. Okay, so what is depolarization? Again, a short summary. What is depolarization? So D refers to reversal of polarity from the polarized state of cell membrane. So it can either caused due to decrease in negativity inside or decrease in positivity outside. What is hyperpolarization? So hyperpolarization means increased polarity of the membrane in the same direction, right? So hyperpolarization. So it can be caused due to increasing negativity inside or increasing positivity outside. So understood about depolarization and hyperpolarization. And what is polarized state? So the state of cell membrane at resting membrane potential is called polarized state. Now we shall study about the effect of various ion channels. Effect of ion channels. So before going deep into the effect of ion channels, first we shall understand the concentration gradient for some of the ions. So from where to where the concentration gradient have been like that we shall understood. So we will be dealing about sodium, potassium, chloride and calcium. So these four are primary ions. So we can know where the concentration gradient is from where to where we shall understand, and then we'll move on to effect of ion channels. First, as we all know, sodium is an extracellular cation, right? So we'll call sodium as extracellular cation. What does it mean? So it has higher concentration in ECF than in ICF. So so the concentration gradient of sodium is from ECF to ICF, right? ECF to ICF. So as sodium is called as extracellular cation, so the concentration is more in ECF, so the concentration gradient is from ECF to ICF. Means the so sodium will move from extracellular fluid to intracellular fluid, which means the sodium will move inside the cell. And potassium, as we already referred, potassium is an intracellular cation, right? So potassium is intracellular cation. What does it mean? So intracellular cation, the concentration of potassium is more in ICF than in ECF. So the concentration gradient of potassium is from ICF to ECF. That means the potassium will move out of the cell. And chloride, so from the Gibbs Donnan effect itself we understood the concentration of chloride is more in ECF than in ICF, right? As we saw in Gibbs Donnan effect, the concentration of chloride in ECF is more than ICF because of protein, right? So where is the concentration gradient for chloride? So same as that of sodium, so it is from ECF to ICF. So as chloride will move inside the cell. And calcium, so calcium is also an extracellular cation, so as the calcium concentration also greater in ECF than in ICF. So calcium also has a concentration gradient from ECF to ICF. So now understood what are all the concentration gradient of sodium, potassium, chloride and calcium. So sodium will move into the cell, chloride will move into the cell, calcium move into the cell, potassium move out of the cell because of its concentration gradient. Now what are the effect of ion channels? Remember this, we'll go into the effect of ion channels. So first what are ion channels? What is an example? What type of transport is of ion channels? So, as we already mentioned in our video on transport mechanism, ion channels are an example of channel proteins, right? Channel proteins. Ion channels are channel proteins. These are example of which type of transport? These are example of selective, simple, passive diffusion. These are example of selective, simple, passive diffusion of transport. So what does it mean? So selective, from the term itself we could understood, so ion channels are selective. Specific ion channels for specific ions are present. So ion channels are selective, and it is simple passive diffusion. So what does simple? Simple refers to it does not require any carrier protein. So that only I stress, ion channels are not carrier protein, they are just channel proteins. So simple, which means they does not require any carrier protein. What is passive diffusion? Passive diffusion, that is along the concentration gradient. Movement along the concentration gradient is called passive diffusion. So ion channels are example of selective, simple, passive diffusion. Okay, so what does ion channels do? Ion channels selectively transport ion along the concentration gradient across the membrane. First we shall see what will happen when sodium channels open. So when sodium channels open, so all these ion channels are present in the cell membrane, right? These are channel protein. They are present within the cell membrane allow for transport of specific ions. So when sodium channel opens, as I already said, ion channels are simple passive, selective simple passive diffusion. So these sodium channels will help in transport of sodium along the concentration gradient. So as we already saw, concentration gradient of sodium is from ECF to ICF, right? So when sodium channels open, what occurs? Sodium influx occurs. What does mean by influx? Flux is nothing but movement of ions, and in means inside. So influx occurs. So sodium will move inside the cell. So what will happen when sodium moves inside the cell? As we know, our cell, this is ICF and this is ECF. This is ECF, and inside is at rest. The resting membrane is at rest. It will be in polarized state, means inside is negative and outside is positive. So when sodium enters the cell, what happens? When sodium enters the cell, as positive charges gets in, the negative charge will decrease, right? This positive charge will neutralize some of the negative charge and will decrease the number of negative charge inside the cell. So there, what will happen then? So there will be decrease in negative charge. So decrease in negative charge. So what is mean by decrease in negative charge? So the decrease in negativity of the cell is termed as depolarization, right? So what is depolarization? Depolarization is reversal of polarity. So it can be attained by two things. One, decreasing negativity inside the cell, or decreasing positivity outside the cell. Here, as sodium moves inside the cell, the negativity inside the cell will get decreased. So which means opening of sodium channel will cause depolarization. What is depolarization? You understood, right? Okay. Then coming on to calcium channels. So when calcium channels open, so what will happen? As calcium channels, as these ion channels are selective simple passive diffusion, the calcium will move along the concentration gradient through this calcium channels. As we know, the concentration gradient for calcium is from outside to inside, ECF to ICF. So what does happen? So when calcium channels open, there will be calcium influx. What is influx? Movement inside. So calcium will move inside. So calcium influx, what does it do? So the same, as calcium is positively charged, it will neutralize some of the negative charges and reduce the negativity inside. So what will happen? So there will be decrease in negativity. So what causes when calcium channel opens? So it causes depolarization. Understood, right? So when calcium channels open, there will be depolarization. Then so when potassium channels open, what does it cause? As potassium channels are also ion channels, they will also transport potassium along the concentration gradient. Where is the concentration gradient for potassium? As we know, potassium is intracellular, so its concentration is more in ICF than in ECF. So its concentration gradient is from ICF to ECF. So potassium will move from the cell to outside, right? So move from in to out. What does happen? So potassium efflux will happen. So flux is movement, F is out, so out of the cell. So potassium will move out of the cell. So what does happen when potassium move out of the cell? So we'll again draw a resting membrane potential, polarized state here. So this is ICF and this is ECF. So inside is negative in the polarized state and outside is positive. Okay, so now potassium is moving out of the cell, right? So when potassium moves out of the cell, positive charges are moving out of the cell, or positive charges is lost from the cell. So what does happen to the negativity inside the cell? It will increase, right? So as positive charges gets outside, the negativity gets increased. As negativity get increased, so this cause increase in negativity inside the cell, right? So increase in negativity inside the cell. What does it mean? Increase in negativity inside cell. What does it cause? It causes hyperpolarization. Potassium channels open, there will be hyperpolarization. Then when chloride channels open, so what does happen when chloride channels open? So the same as the chloride channels, as these are ion channels. So ion channels will allow only simple passive diffusion, right? So selective simple passive diffusion, which means movement of chloride along the concentration gradient. So where is the concentration gradient of chloride? So chloride has a concentration gradient from ECF to ICF. So what does happen? Chloride. So from ECF to ICF, so chloride will move into the cell. So as chloride is negatively charged, the more negatively charges moving inside the cell. What does happen to the inside negativity? So the inside negativity will get increased. So what does it happen? So what does it cause? It causes hyperpolarization, right? So what does, when chloride channels open, what does happen? Chloride influx will happen. As the concentration gradient is from ECF to ICF, there will be chloride influx. So chloride influx will cause what? Hyperpolarization, increase in negativity inside the cell. This causes hyperpolarization. Okay, understood what is hyperpolarization, what is depolarization, and what are the effect of various ion channels on hyperpolarization and depolarization. To understand that, first we should know where is the concentration gradient for. So where is the concentration gradient, and what is ion channels? Ion channels are selective simple passive diffusion transport channels, and they are channel proteins, not carrier protein. We should not confuse channel proteins with carrier proteins. Carrier protein comes under facilitated passive diffusion, right? It's different. So a summary about depolarization and hyperpolarization and the effect of ion channels on depolarization and hyperpolarization. So when does depolarization will be caused? Depolarization. So depolarization is caused by either opening of sodium or calcium channels, right? So either opening of sodium or calcium channels, or it can also be caused by closure of potassium or chloride channels. These conditions can cause depolarization. What are the conditions which cause hyperpolarization? What are the conditions? Hyperpolarization, opening of potassium and chloride channels. So opening of potassium or chloride channels can cause hyperpolarization. Understood, right? So this is a summary of depolarization, hyperpolarization, and the effect of ion channels on depolarization and hyperpolarization. Okay, next we shall see about tonic potential. So what does tonic potential means? What is the definition of tonic potential? So tonic potential is nothing but any change in the membrane potential. As we know, in the rest state, the inside will be negative and the outside will be positive in the cell membrane, and the potential difference existing between this is called resting membrane potential. So any change in this resting membrane potential, till threshold potential is reached is called tonic potential. Tonic potential is nothing but the potential difference when resting membrane potential is changed till threshold potential is reached is called tonic potential. So what is threshold potential? So threshold potential is the minimum potential. It is the minimum potential difference which is required to generate an action potential, or in other words we can say, so the minimum potential difference which is required to fire a nerve. What means firing a nerve or to generate action potential? So means to generate impulse. So the such potential difference change within threshold potential is reached. Is called tonic potential. So tonic potential is the potential change in resting membrane potential till threshold potential is reached is called tonic potential. What does threshold potential mean? So it is the minimum potential difference which is required to fire a nerve, or which is required to generate action potential. What does generating action potential will do? So generating action potential will help in conduction of impulse. So impulse conduction. Only after action potential is generated, the impulse can be conducted, right? Impulse conduction. So the impulse can be conducted. So tonic potential, it's a very easy term, or it's a very small topic. So if for a nerve, so if we give an example for tonic potential, the tonic potential of nerve. So this is a graph which is denoting potential difference across y-axis, potential difference in millivolt across y-axis, and time in second across x-axis. As we already know, resting membrane potential of nerve is minus 70 millivolt, right? It is minus 70 millivolt. And threshold potential of nerve is minus 55 millivolt. So let here it be minus 70 millivolt, and here it will be minus 55 millivolt. So any potential difference change between these two, between the resting membrane potential and threshold potential, is called tonic potential. So understood about tonic potential, right? So it is a small concept. So what is tonic potential? So tonic potential is nothing but a change in membrane potential from resting membrane potential till threshold potential is reached is called tonic potential. Tonic potential is also called as grading potential. Okay, before moving on to the discussion of action potential, let me study clearly about ion channels. Ion channels are nothing but the channel proteins. And these are examples of selective, simple, passive diffusion. So these three pictures denotes the voltage gated sodium channels, and these two pictures denotes the voltage gated potassium channels at different states. So first about sodium channels. So sodium channels, as they are channel protein, they are embedded in the cell membrane. So these proteins, these are channel proteins. The sodium channels have two gates: activation gate, which is facing towards the exterior, and the inactivation gate, which is facing towards the interior of the cell. So when the membrane of any cell is at resting state, that is the membrane potential, when the membrane potential is at resting membrane potential, the inactivation gate is in open state and the activation gate is in closed state. When the membrane potential gets depolarized, as we already said, what is depolarized, what is polarized? So the membrane at which, the state of membrane at which at resting membrane potential is called the polarized state, right? So what is depolarized? Depolarized is the reversal of polarity of membrane from the polarized state is called depolarized state. Means the resting membrane potential initially it will be negative, as it is related. What is resting membrane potential is relative negativity when compared to outside. So initially it will be negative, but when the membrane state gets depolarized, the membrane potential will be getting converted to positive, right? As the depolarized, the polarity will change, so the inside will become positive, so the potential will become positive. So when the membrane gets depolarized, as the negative potential getting converted to positive potential, the activation gate opens. Through which sodium from exterior, as we already said, the gradient for sodium is from ECF to ICF. As ECF has higher concentration of sodium will be drawn into the cell from ECF. So as the activation gate opens, sodium enters the cell. So when the activation gate will be open, so when the membrane is in depolarized state, when the membrane potential is changing from negative to positive side. When it occurs of plus 35 millivolt, when it occurs about plus 35 millivolt, the inactivation gate gets closed. The inactivation gate gets closed and it reaches a state called inactivated state, and this inactivated state will open only when it reaches again the resting membrane potential. So what are the three states of sodium channels? First, it is the resting state. What is resting state? At the resting membrane potential, the activation gate will be closed and the inactivation gate will be open. This is called resting state. And then it comes to activated state. What is activated state? When the membrane potential gets depolarized. So when the membrane potential is depolarizing, the activated gate will open. Both the activated and the inactivating gate are opened in state. This is called the activated state. What is inactivated state? When the membrane potential reach plus 35 millivolt, the inactivation gate gets closed, and this will open only when the membrane potential reach resting membrane potential. So these are the three states of sodium channels. Then about potassium channels. So as the potassium channel has only two states. So why it has two states? As the potassium channel has only one gate which is present towards the interior of the cell. So at resting state of about minus 90 millivolt, or at the resting membrane potential of any cell. So the resting membrane potential for any cell, at the resting membrane potential, the potassium gates, the potassium channel will be closed. And when the potential reach plus 35 millivolt, as the resting membrane potential get depolarized, and when the potential reach plus 35 millivolt, the gate of potassium channel will open and it will allow potassium efflux, right? So as potassium concentration is high in high in ICF than in ECF, the potassium will move out of the cell. And the very peculiar characteristic about potassium channel is that it is slow opening. It is slow opening and delayed closure channel. Slow opening and delayed closure channel. What does it mean? So the opening of potassium channel is very slow and the closure will delayed. So next we are going to a very important concept, which is action potential. So first, about what is action potential. So action potential is nothing but the series of change in membrane potential which occurs after reaching the threshold potential. So when the membrane potential reach threshold potential from resting membrane potential, then the series of changes which is occurring in the membrane potential is referred to as action potential. Okay, why this action potential? This action potential is very important. Only after generation of action potential, the impulse can be conducted. If there is no action potential, impulses cannot be conducted in the nerve. Only after reaching the threshold potential, the series of membrane potential changes in membrane potential will occur, which is termed as action potential. Only when an action potential is generated, the impulse can be conducted. What is meant by conduction? Conduction is the carrying of impulse from one nerve to nerve or within the nerve. Then we'll move on to action potential proper. So action potential, the events we are going to discuss now. So the action potential, so as we already know, for a nerve, the resting membrane potential is minus 50 millivolt and threshold potential is minus 55 millivolt. So when the resting membrane potential reaches the threshold potential, the series of changes will happen in the membrane potential, which is called as action potential. Okay, now first we'll discuss what are the events of action potential. And before that, now we can say that as the membrane potential changes from minus 70 to minus 55 volt, so the graph between these, this is named as tonic potential, right? So what is tonic potential? Tonic potential is any change in membrane potential from resting membrane potential till threshold potential is reached is called tonic potential. So after tonic potential, the series of changes in the membrane potential is actually called as action potential. First, what are the events of action potential? So from this graph itself, we can say first, what is the event of action potential? First event of action potential is that. So from here, the action potential starts, right? Okay, first, what is the event? First event is the upstroke. There is an upstroke here. There is an upstroke in the graph. So first event is the upstroke. So what this upstroke is due to? This upstroke is due to opening of voltage gated sodium channels. Opening of voltage gated sodium channels. So what does this cause? As we already discussed about the effect of ion channels, opening of voltage gated ion channels will cause depolarization, right? Opening of ion channels will cause depolarization. How it causes depolarization? So as voltage gated sodium channels open, we have a sodium gradient from extracellular fluid to intracellular fluid. As at resting state, the intracellular side is negatively charged than extracellular side. As sodium gets in, there will be decreased negativity inside. So decrease in negativity is termed as depolarization, right? So when sodium opening of voltage gated sodium channel will cause depolarization. So how the membrane potential will change? As the membrane potential as in resting state is negative, right? Why negative? So it is relatively negative inside when compared to outside. But when sodium enters in, the negativity will be neutralized by the incoming positive sodium, and the resting potential will develop, will change from negative to positive side. So we are getting a upstroke. Right? Understood? So it minus 55 millivolt, and then it will reach minus 45 millivolt, minus 50, and it reaches up to plus 40 millivolt. It reaches up to plus 40 millivolt. And opening of voltage gated sodium channels will cause depolarization. So the sodium will move from ECF to ICF. So how long this sodium will move from ECF to ICF? Can you understand what I'm asking? So how long this sodium will move from ECF to ICF? So let this be cell. This is ICF. This is ECF. As there is a concentration gradient for sodium from outside to inside, so sodium gets in when the sodium channels open. So how long this sodium will get in? How long this sodium channels? The answer is the sodium will gets in until its equilibrium potential is reached. Until its equilibrium potential is reached. Do you remember, as we already mentioned about equilibrium potential, which is calculated by Nernst equation? Every ions will move only until its equilibrium potential is reached. So what does equilibrium potential mean? There is a different definition for equilibrium potential now. So further, earlier we saw about equilibrium potential. Now we are going to give a different definition for equilibrium potential. What is equilibrium potential? So equilibrium potential is the membrane potential. It is nothing but the membrane potential. Equilibrium potential is the membrane potential at which the electrical gradient cancels the chemical gradient, at which the electrical gradient of an ion cancels the chemical gradient. Electrical gradient of an ion cancels the chemical gradient. This is what a definition of equilibrium potential. So the membrane potential at which the electrical gradient of an ion cancels the chemical gradient. What does it mean? The electrical gradient and chemical gradient. So we shall take this example again. So as the sodium channels open, initially, as we know, inside will be negative at resting state, right? Inside will be negative and outside will be positive. This state is polarized state or in the resting state, resting membrane potential. So when this resting membrane potential reach threshold potential, sodium channels open. So through the sodium channels, sodium will come in, right? So sodium channels comes in. As long as sodium channel open, sodium will come into the cell. So for each sodium entering in, one negative charge will get neutralized. So more sodium, so the influx of more sodium cause neutralization of whole negative charge inside the membrane. Once the whole negative charge get neutralized, the electrical gradient for sodium is end. So initially when sodium channels open, sodium will enter in due to its concentration gradient, and it is favored by its electrical gradient too, right? As it is negative inside, positive outside, so the positive charges gets in. As it is, so the concentration gradient is favored by electrical gradient. But as soon as the electrical gradient gets over, means as soon as the electrical gradient gets depleted, means as soon as all the negative charges gets neutralized by sodium, the electrical gradient is depleted, right? So there is no electrical gradient. As negative charges gets depleted and more and more sodium comes in, sodium inside also becomes positive. So these positive charges will repel sodium charges from coming in. So the electrical gradient depletion will cause sodium influx to be completed. So this potential, so this membrane potential at which the electrical gradient cancels the chemical gradient due to electrical gradient depletion is called equilibrium potential. Now understood what is equilibrium potential? So equilibrium potential. So once again, so summary of equilibrium potential. What is equilibrium potential is that this is the membrane potential at which the electrical gradient cancels the chemical gradient. So what is happening here? First, let us take an example. This is the cell and this is intracellular fluid and this is extracellular fluid. So when the resting membrane potential reaches threshold potential, as initially there will be negativity inside and positivity outside. So when the resting membrane potential reaches the threshold potential, there will be opening of sodium channels, so which cause sodium influx. As the concentration gradient is from ECF to ICF, sodium will cause sodium channels opening will cause sodium influx. As this concentration, as due to concentration gradient, sodium influx happen, and this sodium influx is also favored by the electrical gradient, right? As electrical gradient means as it is negatively charged inside, the positively charged sodium will try to come in. As this negative charge will attract positive charges. So the concentration gradient is favored by electrical gradient initially, but as soon as more and more sodium gets in, it neutralizes the negative charges inside the membrane. It neutralizes the negative charges inside the membrane means at one point of time there will be no negative charges inside the membrane, and the more sodium and accumulation of more sodium ions will cause positive charge inside the membrane, which means so the negative charge is neutralized by the sodium positive charge, and more sodium accumulation will cause increase in positive charge inside the membrane. So as positive charge is developed inside the membrane, the influx of sodium is repelled by these positive charges. So this membrane potential at which the electrical gradient cancels the chemical gradient is called the equilibrium potential. Okay, so this equilibrium potential is calculated by, as we already said, it is Nernst equation. It is calculated by Nernst equation. Nernst equation formula: equilibrium concentration of ion is equal to RT by ZF ln of ionic concentration outside by ionic concentration inside. So in our action potential diagram, so the upstroke will continue. How long this upstroke will continue? The upstroke will continue up to the equilibrium potential of sodium. Up to the equilibrium potential of sodium is reached. Right, understood? So when the equilibrium potential of sodium is reached, the upstroke will stop and it continues as downstroke. So equilibrium potential of sodium is plus 60 millivolt. But even though it is plus 60 millivolt, the upstroke will continue only up to plus 40 millivolt. This is why this is happening. This is due to the sodium channels going into inactivated state. As you remember, as when we see about ion channels, when the action potential reach plus 35, or when the membrane potential reach plus 35 millivolt, the inactivation gate gets closed and it gets into inactivated state, right? So So what happens is that when the membrane potential, even though equilibrium potential of sodium is plus 60 millivolt, when the membrane potential reaches plus 40, plus 35 to plus 40 millivolt, the upstroke stops. Means what stops? Sodium influx stops. Means depolarization gets stopped. Why? Because of sodium channels getting into inactivated state. Means inactivation gets closed. Understood? This is the reason for upstroke. And then the second event is the downstroke. Okay, the second event of action potential is the downstroke. So as we already mentioned, when the resting membrane potential reaches the threshold potential, the series of membrane potential changes will happen, which is called action potential. The first event is nothing but the upstroke. So we saw complete concepts of upstroke. So what is it due to, how long it will take place, and then the second event is the downstroke. So downstroke. So what causes this downstroke to happen? So downstroke is due to opening of voltage gated potassium channel. What is due to? It is due to opening of voltage gated potassium channels. First, clearly understood what does voltage gated potassium channel will cause. For that we'll get from basics. That is, this is a cell with, this is ICF and this is a ECF, and initially it will be negatively charged inside, right? Negatively charged inside with positive charges exterior or to the cell. When the potassium channels open, as we already saw, potassium concentration is more in ECF and low in ICF. So there is a concentration gradient for potassium from ICF to ECF. So there will be potassium efflux. So as potassium is positively charged, loss of positive charge from interior of the cell makes negativity more, right? Makes negativity more. So what will happen? So as the membrane potential due to sodium influx, as it occurs up to plus 40 millivolt. Now as potassium efflux happens, the action potential or the event of this action potential will make the membrane potential to negative. So this is the cause of downstroke. Do you understand? This is the cause of downstroke. And as we already mentioned, potassium channel has a peculiar characteristic, right? What is the characteristic of potassium channel? It is slow opening and delayed closure channel. What makes this slow opening and delayed closure? So what is mean by slow opening and delayed closure? So initially, what is slow opening first? So as potassium, how does this membrane potential reach this threshold potential? We should first understood how does this membrane potential reach threshold potential from resting membrane potential. So the threshold stimulus is the reason. Threshold stimulus is the reason by which the resting membrane potential will reach the threshold potential. What is threshold stimulus? So any stimulus which will make the change in membrane potential from resting membrane potential to threshold potential is called threshold stimulus. Only after reaching threshold potential, action potential can be generated, as that we saw in definition of action potential, right? What is action potential? Action potential is a series of changes in the membrane potential when the membrane potential reaches threshold potential. So how does the threshold potential is reached? Only by giving or applying threshold stimulus. So if we for example, if we consider a nerve, when we are giving a threshold stimulus to a nerve, so the resting membrane potential of a nerve minus 70 millivolt will reach to the minus 55, and then the action potential reach. So as soon as the threshold stimulus is applied, it will cause opening of sodium channels and potassium channels. But there will be opening of sodium channel, but there won't be opening of potassium channel. Why? So as potassium channels are slow opening, so this characterizes the slow opening potassium channels, and the potassium channel will open only when the membrane potential reaches plus 40 millivolt. So potassium channel is actually called slow opening channels. So I repeat, as the potassium channel, as soon as the threshold stimulus is given, the potassium channel should have opened, but it has not happened, right? So only when the membrane potential reaches plus 40 millivolt, the potassium channels are opened. So this is called makes potassium channel to be called as slow opening potassium channel. And this downstroke and as it is a slow opening channel, it opens around plus 40 millivolt, and as potassium will leave out of the cell when potassium channels are open, the membrane potential become negative, and once it reaches the resting membrane potential and it reaches membrane potential of negative value below the resting membrane potential. Why the membrane potential reach below resting membrane potential, more negative below resting membrane potential? As potassium channels are characterized by delayed closure channel, it won't close as soon as the membrane potential reaches the resting membrane potential. As it is a delayed closure channel, the potassium channel will start to close only when the membrane potential reaches more negative below the resting membrane potential. So this is the reason why the downstroke has been extended beyond the resting membrane potential. So once again, summary of the downstroke. So downstroke, when it will be caused, it will start at a potential of plus 40 millivolt. How it is happening and why it is happening? So it is happening due to the opening of voltage gated potassium channel. Opening of voltage gated potassium channel will cause a flux of potassium from cell to outside. So what does it cause? As the cell loses positive charges, its interior becomes negative. So the membrane potential will becomes negative, and as it is characterized by slow opening, it won't open as soon as the stimulus is applied. And also the downstroke will continue beyond the resting membrane potential because of its characteristic of delayed closure. When the membrane potential becomes more negative than the resting membrane potential, the closure of potassium channel begins. So and the third event of action potential is that after hyperpolarization. So after hyperpolarization, so this is minus 70 millivolt, minus 55 millivolt, plus 40 millivolt. So when the membrane potential reaches threshold potential, the series of changes. First there is the upstroke up to plus 40 millivolt, and then there is a downstroke beyond resting membrane potential, and there is next event which is called as after hyperpolarization. This state is called after hyperpolarization. As do you understand? As below as the membrane potential reaches beyond the resting membrane potential to negative, this stage is called hyperpolarization state, right? So the event which happens after this hyperpolarization is called after hyperpolarization state. Okay, this after hyperpolarization is due to delayed closure of potassium channels. How it causes like this? Okay, so we'll again go with our basic diagram. So this is a cell, this is ICF and ECF, and now we have inner negativity, more negative. As it is in the hyperpolarized state, the inner negativity will be more, right? At the end of downstroke, the inner negativity will be more, and outer there will be more positivity. And as we already mentioned, what does closure of potassium channel cause? As potassium channel gets closed, there will be no further movement of potassium from inside to outside, right? As potassium has a gradient from inside to outside, so as the potassium channel has closed, there will be no further movement of potassium from inside to outside, which means closure of potassium channel will cause depolarization, right? As till closure of potassium channel, there will be more and more potassium going out, so the inside negativity will be increasing. But as now the potassium channel gets closed, there will be no movement of potassium outside, so the inner negativity will get increased when compared to outside, right? So relative to outside. So this causes depolarization. So delayed closure of potassium channel is the reason for after hyperpolarization. So after hyperpolarization, there is a small depolarization like graph, right? So this is called after hyperpolarization. hyperpolarization. Why it is towards positive? Why the potential is reaching towards less negative? As closure of potassium channel causes depolarization, which means decreasing negativity, right? So inside negativity it's getting decreased, so which causes the membrane potential to reach again to the resting membrane potential. So after hyperpolarization will cause reaching of membrane potential to the resting membrane potential after the downstroke. Understood? Okay, now we should understood why nature provides us with the slow opening and delayed closure potassium channel. Okay, so first we shall find a reason why there is a slow opening potassium channel. Slow opening potassium channel. Okay, so let us have this an example. Minus 70 millivolt, minus 55 millivolt, plus 40 millivolt. So when the membrane potential reaches the threshold potential, first upstroke, then downstroke, and then after hyperpolarization stage, right? This is what resting membrane potential. When this resting membrane potential will reach the threshold potential, when the threshold stimulus is given, right? When the threshold stimulus is given, or when the threshold stimulus is applied, the resting membrane potential will reach the threshold potential. Okay, so if the threshold stimulus, as soon as the threshold stimulus is given, if the potassium channel gets opened along with sodium channel, as you know, as soon as the threshold stimulus is given, the sodium channels will open, right? So sodium channels will open, and when it reaches the resting threshold potential, more and more sodium channels open and cause upstroke. But normally when the resting membrane potential, at the resting membrane potential, when the threshold stimulus is open, sodium channels will open and cause the membrane potential to reach threshold potential, and when the threshold potential is more and more sodium channels gets opened in a positive feedback cycle, right? Positive feedback cycle, more and more sodium channels will open, and it will cause the and it causes the upstroke. But if the potassium channel is not slow opening, when as soon as the threshold stimulus is given, if the potassium channel also gets opened, so we'll come to our basic diagram. This is ICF, this is ECF. So as sodium channels open when the stimulus is given, sodium will come into the cell, right? As the concentration gradient is more outside, so the sodium will. And as simultaneously, if the potassium channels are also open, the potassium will go out, right? So the commencement of positive charge and the leaving positive charge from the cells will be neutralized. So there will be no upstroke, or there will be no attainment of threshold potential is possible, right? So only if the sodium channel is only opened, the threshold resting membrane potential can reach the threshold potential with the help of sodium influx. But if the potassium channels are also open, the sodium influx and the potassium efflux will occur simultaneously, and there will be no change in membrane potential. So nature provides the slow opening potassium channel. Okay, this is the reason why the potassium channel is slow opening, and the reason why the potassium channel is delayed closure channel. So if the potassium channel is normal closing channel, as soon as the downstroke reaches resting membrane potential, if the potassium channel closes, the potassium channel close will cause what? It causes depolarization, right? So it causes depolarization. So the membrane potential, as soon as reaching the resting membrane potential, if the potassium channel open, the membrane potential will again reach up to threshold potential. Right, as depolarization happens, there should be less negativity of the membrane potential. So if the potassium channel is normal closing or closing promptly when the membrane potential reaches resting membrane potential, the membrane potential will again reach less negative value, and it can reach up to threshold potential. So it can generate next action potential. But when the membrane potential reaches resting membrane potential, if the potassium channels have closed promptly, then the closure of potassium channel, which cause depolarization, will make the membrane potential to become less negative. So it may cause the membrane potential to reach the threshold potential and can generate the next action potential. As this shouldn't happen, nature provides us with delayed closure potassium channel. So now the summary of action potential. summary of action potential. So first, about what is action potential. Action potential is nothing but a series of change in membrane potential once the resting membrane potential reaches the threshold potential. Okay, so what we have three events, right? What are the three events of action potential? First is upstroke. What upstroke is due to? Upstroke is due to opening of voltage gated sodium channels, right? Voltage gated sodium channels. What does it cause? It causes depolarization. So the membrane potential becomes more positive, will go towards positive. And it how long will the influx of sodium will continue? Once when it reaches the equilibrium potential of sodium influx will stop. So equilibrium potential of sodium, when equilibrium potential of sodium is reached, the sodium influx will stops. Okay, so equilibrium potential of sodium is plus 60 millivolt, but the upstroke stops at plus 40 millivolt. This is due to as sodium channels at range of plus 35 to plus 40 millivolt, it will undergo inactivated state, right? As we already saw in the state, right? Inactivated state. So upstroke will continue only up to plus 40 millivolt. Then second event is the downstroke. What is second event? Is the downstroke. What downstroke is due to? So downstroke is due to opening of voltage gated potassium channels, right? So opening of voltage gated potassium channels. Potassium channels are characterized by slow opening and delayed closure. As potassium channels open, all the potassium from inside of the cell will go outside. Then the inside negativity will increases. As the positive charges are lost from the cell, the inside negativity increases. So the membrane potential reaches negative, and it go beyond the resting membrane potential. It goes beyond the resting membrane potential. Why? So why this goes beyond the resting membrane potential? As potassium channels are characterized by delayed closure channel, the downstroke goes beyond resting membrane potential. And third stage is the after hyperpolarization. So why does after hyperpolarization state happens? Due to delayed closure of potassium channels, right? So as closure of potassium channel causes depolarization. As we already saw in the effect of ion channels, closure of potassium channels causes depolarization, which means the membrane potential will become less negative. So this is the after hyperpolarization state. These are a summary of action potential. Now let us see the last topic of today's discussion, which is conduction of impulse. Okay, so as we already mentioned, only after generation of action potential, impulse can be generated. So for generation of action potential, we need threshold stimulus. Threshold stimulus is nothing but the stimulus which can make the membrane potential from resting membrane potential to threshold potential, right? Threshold potential. So only after action potential is generated, the impulse can be conducted. Okay, so let us take the example. This is nerve fiber. So this portion of nerve fiber is initially as we know, the inside of the membrane will be negatively charged and the outside of the membrane will be positively charged. Okay, as we know this. So first, if the stimulus is applied to this point, consider if the stimulus is applied to this point, how the polarity will be changed here. So now this will be nerve fiber. The nerve fiber or inside will be negative and outside will be positive, but the place where the stimulus is applied will be reversed in polarity, right? So inside will be positive and outside will be negative. And this is the next place, this place where inside will be negative and outside will be positive. So when the action, so this is this place where the action potential is generated, right? This is the place where action potential is generated. So after generation of action potential, as there is positive charge inside and negative charge outside, the positive charges will flow along its both sides. Positive charges will flow along both sides. So the flow of positive charges will make, what does it make? So it makes this area to get depolarized, right? Depolarized in the sense this flow of positive charges to this state make this resting membrane potential to reach the threshold potential. So you understood, right? So makes this area's resting membrane potential to reach the threshold potential. So what happens when the resting membrane potential reaches the threshold potential? The action potential will be generated here. So by this action, the whole impulse, whole stimulus is conducted along the nerve fiber. So this is the pathway, or this is the mechanism by which the impulse is conducted. So what is happening? So when the point where the stimulus is applied will generate action potential initially. So after action potential generation, the inside will become positive and the outside will become negative. So what happens? The inside positive charge will flow towards the adjacent sides, making the adjacent negative charge to reach somewhat positive, which means it makes the adjacent area of membrane to make the resting membrane potential to threshold potential. So after threshold potential is reached, action potential is generated. By this, the conduction of impulses happens. So this is inside, and outside the same will happen. What happens? The positive charges will flow towards the negative charge and will make a action potential to generated. So by this mechanism, the impulse are conducted. This is actually called circular current flow mechanism. Circular current flow mechanism. As positive charges flow, so the flow of positive charge is called current, right? So this is the circular current flow mechanism by which action potential is generated. So now the direction of impulse conduction. So if this is the nerve fiber, consider this as the nerve fiber. So if this is the nerve fiber and this is the axon cell body. The cell body, this is axon and this is axon terminal. Axon terminal. So if the impulse is generated in this part of this neuron, if the impulse is generated in this part of the neuron, this impulse can be transmitted in both directions. Very important. So the impulse can be transmitted in an axon in both directions, that is towards the cell body and also towards the synaptic terminal. So axonal conduction takes place in both directions, that is both towards orthodromic and antidromic direction. So now what is orthodromic and what is antidromic direction? So the direction of conduction towards the synaptic terminal. So the direction of conduction towards the synaptic terminal is called orthodromic conduction. And the conduction which is opposite to this direction, means towards cell body, is called antidromic conduction. Antidromic conduction. So about conduction of impulse, as soon as the impulse is generated, as soon as an action potential is generated, the positive charges flow towards the adjacent sites and makes that site resting membrane potential to reach threshold potential, so that action potential generate in that area too. So by this mechanism, the action potential or the impulse is transmitted towards both sides of the nerve, and the mechanism is circular current flow, and the action potential is transported in axon in both directions. So axonal conduction is bidirectional. What does it mean? Axonal conduction is bidirectional, that is both towards the end of synaptic terminal, which is orthodromic conduction, and towards the cell body, which is antidromic conduction. This finishes our discussion on electrophysiology. The handwritten notes of these topics are given in our Telegram channel, the link of which is given in the description. Thank you.